Hey everyone, we'll start in 2 more minutes. Give people time to find that new link. I'm playing this because when I played music yesterday or the other day, the copyright filters went off. If you play a song that's before 1924, it's public domain. So this song is older than that. Yeah, pretty sure he's copyrighted. I even played some royalty-free music, and it just said that the reason why I had to start a new stream because I was playing royalty-free music. What it claimed was royalty-free music, and YouTube said there was a copyright claim. So, yeah. Sorry, it scares you, Corey. Good morning, everyone. Welcome. Thanks for tuning in. I know you all took the mini quiz. Let's go over it. Let's go over it. The grade is not final. Your mini quiz grade that has been automatically generated is not final. I need to go through it again and check your spelling. So many of you will get full credit for a given question if you just added an S to make it plural, for instance, or you can get half credit if you're off by more than a letter, but I still recognize what it is. I can come to school. They can't pull me away from here. Developing online classes, that's considered essential. So I got the whole building to myself. Well, me and I think his name's Scully, her, they. This one's name is Scully, I guess, because that's the only name I could come up with that was a good pun. Scully. If you have a better name for Scully, or if you have a hat or clothes that Scully can wear, let me know. So we'll go over the mini quiz. We'll go over the brain. I've got lots of brain models that we'll go over in lab. A lot of brain models that we'll go over in lab, and we have human brains. So we'll go over it. I don't know if I'd call this a weave. It's more like a lace front. It's actually what this actually is. This is friend got me as a prank, not a prank, but like a gag gift. Remember when man buns were that in thing? And I tried wearing it, but it's too thick, and my hair is not the same thickness, so it wouldn't hold. But this is like one of those fake man bun things. Well, Corey, you come up with a better one. Right, lots of stuff in lab. Lots of cool brain stuff in lab. I guess they're still in. I can't wear it. Lab check 8. Remember the new way that you're doing this for histology. You are finding pictures somewhere online, finding unlabeled pictures and labeling it yourself. Have a version that's unlabeled with leader lines, and then a version that's labeled with the filled in thing. That way you can test yourself when it comes time for the practical, which is next week. Your practical is in 1 week. It will be online. You also have that activity 7 workout plan due next week. And then your quiz on Monday, which is also online. It's shortened. Normally it's 40 minutes. I think it's going to be 35 minutes now. So, and it is open note. So that's why I'm shortening it, because it's open note. But that does not mean that you should not prepare. You need to know your stuff, because you're going to have to go quick, and yeah, you don't want to spend all your time just looking through your notes. If you forget a spelling, that's what it's there for. You should know exactly what page to turn to if you need to turn to something. So just because we're open note does not mean you should not study. You should study just as much, if not more. If you have any DSS requirements, have the DSS office email me, or take a picture of your whatever sheet, whatever what's it called? What is it called? Accommodation sheet. There we go. If you have an accommodation sheet, or if you can get a DSS counselor to email me, that's all I need. Just get me that, and I'll work with you. If the time limit runs out and you don't press submit, will it take your answers, or do you get a 0? That's a great question. I don't know. If that happened to you, I will double check. Email me a message saying that happened to you, and I'll double check to see what's going on. I'm still I'm still learning about this. When will your practical start? Check the latest announcement. Well, not the latest announcement, the latest informative announcement. Practical for Monday, Wednesday starts at 11:15, and for Tuesday, Thursday, it starts at 8:30. You can sleep in a little bit. Any other questions about what we're doing today? Questions? Okay, let's review the quiz real quick, the mini quiz. First question, I'm asking you for the region of the neuron. In this picture, we're looking at, you can see skeletal muscle, longitudinal section. Here are axons of a neuron, and then here's the end of the neuron. This is the axon terminal. Axon terminal is answer for the first one. Question 2, what do you call an axon pathway that goes from sensory to central nervous system? So here's the dendrites of a sensory neuron towards the spinal cord, for instance. This pathway, it's approaching. This is an afferent with an A. So we had axon terminal, afferent, pathway. Question 3, what do you call the region of a neuron where we decide whether or not the action potential is going to happen? You have a high concentration of voltage-gated sodium channels here. Tamana, again, I'm going to go through and double check your answers. You'll get full credit for that. I need to go through these again. Your first pass of the mini quiz grade is not official. I need to go through it. For question 3, this is the axon hillock. Question 4, which glial cell? Here you can see the cell body, and it has multiple extensions that form myelin sheaths. If you have a glial cell that forms multiple extensions, multiple wrappings, that's an oligodendrocyte. If it's only one wrapping, one cell per wrapping, that would be a Schwann cell, but this is an oligodendrocyte. And here we are looking at cells lining a ventricle, a cerebrospinal fluid-filled space in the nervous system. The glial cell that lines it is the ependymal cell. Psi, the difference between axon hillocks and nodes of Ranvier. Axon hillocks, let me go back. No, that's not the best picture, hold on. The axon hillock is the region between the cell body and the axon. So it's the region right between the cell body and the axon hillock. This is our decision point of whether our graded potential will activate an action potential. You do find voltage-gated sodium channels here. At the nodes of Ranvier, they're past the cell body. They're in between myelin sheaths. You do also find high concentrations of voltage-gated channels. So in that way, axon hillocks and nodes of Ranvier are similar. But the biggest difference is that the axon hillock is the first one. Think of it as the first domino. Have you pushed that domino hard enough to cause a chain reaction in the rest? Or that first one is the deciding one, the axon hillock is the deciding one. Once again, Jose, if I need to double check the answers, so I will I will go back and grade them. Any other questions on these mini quiz things? Okay. Okay. Any last things before we start with the brain? How can you tell the difference between myelin sheath and lining? Myelin sheath, and just a reminder, the reason why it takes a minute from you asking questions and me answering them, there's a bit of a lag. So yeah, expect a 15 to 30-second lag. Myelin sheath versus, what was it? Myelin sheath versus lining. Sorry, Corey. Myelin sheaths, let me show you a different picture. I'm opening up the nervous tissue lecture so I can show you myelin sheaths versus linings. If we're looking at histology, myelin sheaths are always surrounding an axon. So this little dot is an axon in cross section, and you can see the myelin around it. There's always going to be stuff around it. There's stuff surrounding the myelin and everything else because it's packed in with the nervous tissue. This is in cross section. Here's myelin in longitudinal section. Again, you just have stuff all next to each other. If we look at ependymal cells, where's my better picture of this? I guess we'll have to. If you're looking at ependymal cells, there's always like a white space. If it's the spinal cord, it's the central canal. If it's the brain, it's in a ventricle. And we're going to go over what a ventricle is. I'm going to go forward, but if you still have questions, because again, there's lag here, I'll go over it. Will I specify whether we're in the peripheral nervous system or central nervous system? Sometimes, yes. If it's really hard to tell, I will specify. But for example, when I showed you dorsal root ganglion, the other day, you should know that that's peripheral. So in some cases I will, but if there's enough cues for you to tell that whether you're in central or peripheral, I won't. So one example is dorsal root ganglion, you should know that's peripheral. If I show you a model, like that model of the neuron, you should know that's a motor neuron, so the cell body's in the central nervous system, but its axon is peripheral. And thank you for helping each other out in the chat. Okay, you ready? Can you make a video differentiating the two? The two types of cells, two types of nervous systems. I believe I made that distinction in yesterday's videos and in the separate videos where I just go over the histology and where I go over the histology and everything. I can do that in lab. I'm happy to do that in lab, Ashley. Go over the peripheral and central nervous systems. But if it's in the brain or spinal cord, like if we see pyramidal neurons, Purkinje cells, then we're definitely in the central nervous system. If we see dorsal root ganglion, or just a nerve, like a peripheral nerve that's been cut, then we're in the peripheral nervous system. All right, let's get started. Looking at the brain, we're identifying not just brain structures. The chat will be visible for both lecture and lab. Sarah, can you see what on the slide? We'll be going over structures, spaces, and fluids that protect and support the brain. So we're not just going over brain regions, but we're also going over all the things that help support the brain. We'll go over the different areas of the brain, of course, and then we'll eventually get to the different cranial nerves. That'll be next time. Are we going to see peripheral nervous system and central nervous system on slides? Sarah, do you mean histology? The lab chat wasn't visible. For Sarah, your question about peripheral nervous system, central nervous system, histology, once again, it's like if you see pyramidal cells, if you see Purkinje cells, if you see a spinal cord, then you know it's central nervous system. If you see a peripheral nerve with a different connective tissue surrounding it, if you see dorsal root ganglion, then you know that that's peripheral. Okay, I see. The chat wasn't visible. I don't remember unchecking it, but I'll double check to make sure that it's visible. We're doing the best we can, Corey and Victoria. I know it's not perfect. Would you rather have a video chat? Because we could do that too. Like I could have it so that you video in and then you can speak as well. If that's something you'd like, go ahead and say it in the chat. I can set that up for next time. All right. We are looking at a model of a patient's injury. This patient's name is Phineas Gage. If you've ever taken a psychology class, you've probably heard his name because he's a classic case study. He's a railroad worker. I forget from what year, but probably early 1900s, maybe late 1800s, back in the day. And he was, when you work railroads, you work with dynamite and steel rods or whatever kind of rods, and this rod went through his skull. You can see the anterior side of his brain, going through his eye, his cheek and eye. It went straight through his brain, and it damaged his brain, obviously. He had lots of brain damage, but it's only in this front area, and he survived. Like his overall health was fine. What was different though was his personality. So what this tells us is that different parts of the brain are going to be useful for different things. The front of the brain, it's not vital for survival, but it's important for other things. He became really, really rude, really unpleasant to be around after damaging the front of his brain. He was not that way before. So we'll take a look at different regions of the brain, what they're generally important for, and which ones are essential for survival and which ones are for making us, who we are. So when I use the term brain, we're talking about the center of our nervous system. I mean, the spinal cord is part of the central nervous system also, but even the spinal cord leads up to the brain, leads to and from the brain. It's way, way, way more complex than the spinal cord. 20 billion neurons or so, give or take. It allows for very quick, coordinated responses to changes in your environment. So if someone throws a ball at me, I can quickly respond and catch it. If I get cold, I can immediately start shivering. Very, very quick responses. It does require lots of protection and support. Being arguably our most vital organ, It requires a lot of protection and support. We've gone over our cranial bones. You should know what they are. You should know frontal, parietal, temporal, occipital, sphenoid, and ethmoid. Those are all part of the cranium. They're there to protect the brain. That's one of their major roles. You should know, we will be going over the different meninges and fluids. Here's a fun word, meninges. Where's my chalk? Meninges, that's plural. Singular is meninx. Just a heads up. Plural is meninges, the protective covering of the brain. Singular is meninx. It's a lot like phalanges. Well, if we're talking about all of your phalanges, all of your fingers or toes, that's plural. Singular is phalanx. I never know how to pronounce this, but it's with an X. Just a fun plural rule, singular plural rule in, shoot, is this Latin or Greek? I think it's Greek. I don't know. One of those. All right. You remember the very first day I showed you a picture of a brain and then showed you neurons? We're making that transition. How do these very small things, cells, very branched, form something like this? How does their structures relate to their function? What does the meninges do? It's a covering protecting the brain, and we're going to go over the different layers. Everyone in the chat, do me a favor. Avoid using pronouns like it, because I don't know what you're referring to. Thank you. Yeah. So, meninges. Meninges are layers of membrane. Not the same as when I use the term membrane, I'm not referring to the same thing as like mucous membrane or things like that. It's just a covering. Layers of membrane surrounding the brain and spinal cord. So it covers both. There are 3 layers: Dura mater, arachnoid mater, and pia mater. The word Dura means like durable. If something's durable, it's tough. The Dura mater is a very tough outer layer covering the brain and the spinal cord. It's the most superficial. So if we're looking at a superior view of someone's skull here, a cadaver skull, you can see the skin. This is the scalp. If you peel away a first layer, you see the aponeurosis, that epicranial aponeurosis. Peel that away. You can see the periosteum of the skull. So this would be like the frontal and parietal bones. Take that away. You see more bone. Take that away. You can see the dura mater. Dura mater is the tough outer layer. I'll be showing it to you in an actual brain. You'll see how tough it is. It's very, very tough. In between the dura and the next layer down after dura mater, it's called arachnoid. It's a very thin layer. It looks like a very thin, clearish membrane. It's called arachnoid. Maybe this reminds you of a spider, like arachnid. It's a web-like network of membrane. That's why it's called arachnoid. We'll see why that's important. Of course, as always, structure relates to function. And then pia, the word pia means soft. So this is the very thinnest, softest layer, outer covering of the brain. You can't really see it. It's so thin. So you have to assume that it's a very, very thin layer that's covering every single groove of the brain. The arachnoid mater does not go with every single groove. The pia mater does. So whenever you see a brain where you see all the grooves and wrinkles, just assume that it's covered by pia mater. We don't have the tools to peel it off like we do with these other meningeal layers. You get to see the brain and layers on our cadavers. We already have brains that have been removed from other previous cadavers, and yes, I'll be showing that in lab. All right. Okay, so let's say we're looking at a person that's a mid-sagittal view of a person. So here we have, here's the brain. This is anterior. This is posterior. Here's the brain, and the brain is continuous with the spinal cord. So here's the brain. Here's the spinal cord in yellow. Read slide 5 for meninges. Oh. So what I've drawn here in yellow, that's nervous tissue. Meninges is surrounding nervous tissue. So if we go in order, first layer is the pia mater. I'm going to make a key over here on the side. Next is arachnoid mater. And arachnoid mater is kind of web-like. So I'll show you what I mean by that. It takes up a bit more space than the pia mater. And then dura mater is on the very outside of that. So the nervous system, the central nervous system, is very protected. 3 layers. 3 layers of meninges covering up the central nervous system, brain and spinal cord. Okay, I'll get back to this in a minute. First, let's go over the dura mater. The dura mater is the thickest, it's the most superficial, and it's divided into 2 layers. One layer is continuous with bone. It's continuous with the periosteum of bone, so that's called the periosteal layer. Remember our skull, just underneath, just deep to the skull, so just deep to the bone, that's where the meninges is. So here's zooming in on that. Here's bone. Here is the periosteal layer. Periosteal layer. The one closest to the arachnoid, that's called the meningeal layer. Sometimes they're fused together. Sometimes you have the periosteal layer and the meningeal layer fused together, but sometimes they separate and they provide a space. This space is so that a blood vessel can fit through it. The term for a large vein is a sinus. Where is it? The term for a large vein is a sinus, a venous sinus. Venous for vein. So this space is for a dural venous sinus, a large blood vessel that goes through the dura mater. So let me draw that on my diagram. Again, sometimes the periosteal layer and meningeal layer are together, but sometimes they separate. So let's say here's the periosteal, here's the meningeal, and then in between, this is a blood vessel, a dural venous sinus. Here's a model of a mid-sagittal cut of a skull. You can see here's the bone. And then just deep that, this white line, that would be the periosteal layer that's forming the periosteum of the bone. This next white line is the, let me get my pointer. Okay, periosteal layer. Here's the dural venous sinus going all the way along this middle part. And then this next white line next to the brain, that's the meningeal layer. You can see this dural venous sinus goes all the way mid-sagittally. So there are only so many of these. We're not going to go over every single one. I just want to show you a good example, but there is one that goes that's in the middle of the skull, like this. This is a vein that drains blood away from the head, a dural venous sinus. Sinus is just a term for a large blood vessel, a large vein. Dural venous sinus. That's one model where you can see it. There's one more model. Here's a clear outside model so you can see the interior parts of a brain, and you can see here's a dural venous sinus. This very not as clear hard plastic, that's representing dura mater. And then you can see this is a dural venous sinus going in between. Cool. So, in between, so you might know that we have two hemispheres for our brain. The dura mater doesn't just cover the outside of the brain, it also goes in between the brain. It doesn't go in between fully, it just goes in between partially. So again, looking at our mid-sagittal cut, our brain does connect here. We'll go over that connection later, but somewhat separating the brain, so like partially mid-sagittally, we've got this dura mater in the middle. So if you can imagine, there's dura mater, like there's dura mater over here, there's dura mater over here, the two fuse together and they pass in the middle here, but only so far. They only pass, see this curve here? This is a connection between the hemispheres. The fused dura mater connects in between here. The term for that is the falx cerebri. Falx means like a hook shape, like a crescent shape. So this is a crescent shape. Cerebri, this area of the brain, the outer wrinkled part of the brain is called the cerebrum. So falx cerebri, it's a crescent shaped thing in between the cerebral hemispheres. The term for this mid-sagittal cleft between the hemispheres, that's called the longitudinal fissure. So it fits within the longitudinal fissure of the brain. Remember the crista galli? Who can tell me what bone the crista galli belongs to? The falx cerebri attaches in the front to that crista galli. Anyone? Anyone? I know there's a lag. There we go. Ethmoid, yeah. We're learning all these bone features for a reason. Here's yet another reason why we need to know them. The crista galli, that's what attaches the falx cerebri on the anterior side. There are other parts like falx cerebelli and tentorium cerebelli. We're not going to go over those terms. I do want you to know falx cerebri though. We will be able to see it on the cadaver brain. All right, next layer deep, next layer down, the arachnoid. If this is the periosteal layer of the dura, here's the meningeal layer of the dura, and then here is a dural venous sinus. Deep to that, shown here, that's the arachnoid membrane. The arachnoid membrane. It looks all web-like. Because it's web-like, that means there's space here, and that space is filled with fluid. It's called the subarachnoid space, filled with cerebrospinal fluid. So ventricle, we've mentioned ventricles before. I know we haven't defined it yet, but we'll get there. Ventricles are filled with cerebrospinal fluid. The central canal is filled with cerebrospinal fluid. So is the subarachnoid space. Here's the membrane, and then there's a space below it. That's filled with fluid. It has what forms these web-like things. That's collagen attaching it, securing it down to the pia. Our diagram here, we can label like this space here in between. That's the subarachnoid space. Where's the longitudinal fissure? It's you can't see it on this because this is mid-sagittal. You'd have to see it from this view. It's this split. That's a longitudinal fissure. Right. Why do we need this subarachnoid space? I mentioned how the cerebrospinal fluid is kind of like really ultra filtered blood. We definitely don't want viruses and bacteria to infect our brain. You may have heard of meningitis. That's when the meninges doesn't do its job. That's when you get an infection in this area. So when it is working properly, we're preventing, the cerebrospinal fluid is preventing things from coming in with just really ultra highly filtered blood. There's still lots of oxygen, lots of nutrients, just we're minimizing pathogens. Just like blood, we need to circulate blood. We also need to circulate cerebrospinal fluid. So as cerebrospinal fluid flows through the brain and spinal cord, nutrients are getting taken away, waste is being added. We need to get rid of the waste and then return that waste to the bloodstream. This subarachnoid space is really, really important for returning waste back to the bloodstream. That's why you have these venous sinuses. And I'll show you the flow in a minute. Pia mater, pia mater, that's the deepest, the thinnest. If you see the brain and you see all these folds, the pia mater goes directly with it. It follows every single fold, they're called gyri and sulci. Oh yeah, there it is. The term for a wrinkle, where's my thing? The term for a wrinkle is a sulcus. The term for a wrinkle is the sulcus. So the thing for what bulges out, that's a gyrus. Just like you have a peak and a valley, a peak would be a gyrus, or plural gyri. A depression, a valley, would be a sulcus, or a plural sulci. I'll type out the singular and plural. Whoops. The pia mater, the way it attaches to the brain, it's anchored by astrocytes. It's anchored by astrocytes. So there's another function for astrocytes. Helps anchor down the pia mater. Blood vessels also get anchored down between the pia and arachnoid. Blood vessels get anchored down between the pia and the arachnoid. So that brings us, we know that the astrocytes are important in holding down the pia. The astrocytes form that blood-brain barrier. It's lining most blood vessels that supply the brain. I say most because there are some exceptions, and we'll get to that later. But most of the brain is covered, most of the blood vessels in the brain are covered by astrocytes so that it can regulate the passage of molecules. We don't want pathogens and toxins to come into the brain easily. The way that these astrocytes can regulate what's going on, it's covering up the blood vessels and forming tight junctions. We have tight junctions here to prevent leakage of stuff from blood vessels out into the brain or spinal cord. So astrocytes, and here's a longitudinal cut. Astrocytes cover up blood vessels, ensure that nothing's leaking out. What do you want me to say again? Keep the questions coming. Is the blood-brain barrier the pia mater? Not exactly. The pia mater and blood-brain barrier are related, but the pia mater is the membrane covering, and as the astrocytes anchor the pia mater down, they also help form the blood-brain barrier. So, Mackenzie, no, not exactly. More the astrocytes form the blood-brain barrier, and the astrocytes also anchor the pia mater down. Okay, back to that cerebrospinal fluid. So protecting the brain, we have the meninges, but fluid can also help protect the brain. If you have, like one reason why things are packed in fluid is that if you're in water, the water is gonna take the impact. So cerebrospinal fluid takes the impact. It's a cushion. It acts as a cushion. So that's one really important role of cerebrospinal fluid. It acts as a cushion, and it transports nutrients and waste. So we need circulation, and there is circulation. I'm pretty sure I have an animation of this later. But the fluid in the ventricles, in the central canal, in the subarachnoid space, it does circulate around. Remember, cerebrospinal fluid is produced by ependymal cells. Where do we find ependymal cells? It's lining ventricles and central canal. Here's a space within the brain, and let me show you. Here's a mid-sagittal cut of the brain. This space here, this is a ventricle, and see this red stuff? That's representing choroid plexus. That's representing choroid plexus. Choroid plexus is where you find ependymal cells that produce cerebrospinal fluid. So zooming into this choroid plexus, here you can see a blood vessel, and then over here you can see ependymal cells, and then here is the cerebrospinal fluid. Blood can get filtered by these by these ependymal cells, and then that filtered fluid can come out here. So we're only taking the good stuff. This is how cerebrospinal fluid is formed. It's formed by choroid plexus. If we're forming fluid, we also need to get rid of fluid. That's how circulation works. It needs to keep going in one direction. Is cerebrospinal fluid affected by vertigo? Vertigo is dizziness. That is something to do with your inner ear organs. We'll get to that in a couple of weeks. Here is a ventricle. There are several ventricles. We'll get to them later. Here's a ventricle with choroid plexus. This is where cerebrospinal fluid is produced. Cerebrospinal fluid is produced by the choroid plexus, by the ependymal cells. Cerebrospinal fluid can flow through the ventricles, can flow through the central canal because it's all connected. Notice I'll get to the CSF cavity in a second. CSF goes through the ventricles, through the central canal, and then it exits out the very tip of the spinal cord. And then we can get CSF. Once we exit the spinal cord, now we're in subarachnoid space. So we can go this way or this way, whichever way, come out. And then the way we exit from the subarachnoid space, there are these protrusions that go into the dural venous sinuses. Those protrusions are called arachnoid villi. CSF is reabsorbed into the venous circulation by arachnoid villi. Okay, there's a lot going on here. Let me show you on our diagram over here. All right. So we have several ventricles in our brain, several fluid-filled spaces in our brain. So here's a ventricle, here's a ventricle. I'm not drawing it perfectly, but it'll do. Ventricles have choroid plexus, the stuff that produces cerebrospinal fluid. So it produces the cerebrospinal fluid. Cerebrospinal fluid is produced. It circulates through the ventricles of the brain. It circulates down to the central canal. Circulates to the subarachnoid space. So now we're in the subarachnoid space. Going through the subarachnoid space, through the subarachnoid space, subarachnoid space. And eventually we need to get rid of it because by this point, all this cerebrospinal fluid has lost nutrients, has lost oxygen. So we need to get it out and bring fresh CSF in, brings fresh CSF in from the choroid plexus. So the way we get rid of cerebrospinal fluid are through these little protrusions called arachnoid villi. What color should I use? Running out of color. Green. Okay. So this in orange, that's the arachnoid mater. In orange, that's the arachnoid mater. The arachnoid mater makes little protrusions. I guess I should use orange. The arachnoid mater makes little protrusions, little finger-like protrusions that extend into the dural venous sinuses. The arachnoid villi, the arachnoid villi are these extensions that go into the dural venous sinus. In that way, any fluid, any CSF that's in the subarachnoid space can now exit and drain to the dural venous sinus. We're getting circulation. We start at point A, choroid plexus, go out, and circulate out around. That way we can circulate fresh CSF, get rid of old CSF. CSF is created by the ependymal cells of the choroid plexus. It's removed by arachnoid villi into the dural venous sinuses. On our diagram here, you can see there are arachnoid villi all over. So here are two arachnoid villi that are shown here. Arachnoid, did I write down arachnoid villi? I should spell it out for you. But here it is zoomed in. Here you can see the arachnoid membrane, arachnoid mater, and you can see the branching of the arachnoid mater, the subarachnoid space. And this protrusion into the dural venous sinus, this protrusion, that's what is draining fluid. We're draining CSF into the dural venous sinuses. What happens to old CSF after it gets drained? It's back into the blood circulation, and so now it's a waste that our body needs to process. Livers can get rid of old waste. We can urinate stuff out. We can reoxygenate it by getting it to our heart and then getting it to our lungs. So we're treating it just like any other venous blood. Any other blood that's in your veins, same thing happens to it too. Narcissus, did that clear up CSF cavities? On our model here, this very, very tough outside is representing dura mater, or sorry, this is representing brain. Sorry, not brain. Okay. This very, very tough outside, it's representing bone. There we go. We know it's representing bone. You can see the external occipital protuberance. The periosteal layer you can't really see, but then this is the meningeal layer of the dura mater. And then here is the dural venous sinus. These little things, those are representing arachnoid villi. Those little things are representing arachnoid villi. And then this hard plastic, you can see that it's not perfectly going with every groove. Like you see some grooves, but it's just kind of covering it. This is representing the arachnoid mater. Pia mater would just be directly against the brain. So once again, arachnoid mater, arachnoid villi, dural venous sinus, dura mater, skull, bone. Any other questions about meninges and CSF flow? I'm going to drink water real quick. Okay. Oh, let me just show you this real quick. I didn't really point this out. On this picture here, this is if we cut our skull this way, a horizontal cut, so we're looking inside at the base of the cranium. You can see this little line, that's the falx, part of the falx cerebri. And you can see how we form a split here, and here's space for a vein. This is an example of a venous sinus. So here's one sinus, here's another sinus. So arachnoid villi will be draining into these. Okay. So what are these ventricles? What are these spaces in the brain? In this picture, what's shown here in blue, you have to imagine that it's filled with fluid. So this is not solid material. These are spaces that are filled with fluid. That's what a ventricle is. Fluid-filled, CSF-filled spaces, cerebrospinal filled spaces within the brain, where you also find that choroid plexus. Jose, if you want me to break something down for you, let me know. Let me know. Okay, there are 4 ventricles. There are 4 ventricles. The biggest ones are the lateral ventricles. There are 2 of them. You could technically call them the first and second, but yeah, you just call them the left and right lateral ventricles. Here's our brain, mid-sagittal cut, so you're only really seeing 2 dimensions. So if you could see it in 3 dimensions, this space here should be 3-dimensional, and that's what's shown here in blue on our diagram. This model is representing that. This model here is representing the fluid-filled ventricles of the brain. There we go. Yeah. Okay. So you have a left lateral ventricle and a right lateral ventricle. You can see how long it is, how it curves around. Just this one piece is one continuous ventricle. Left lateral ventricle, right lateral ventricle. See the pink on the underside? That's representing choroid plexus. You've got choroid plexus and other ventricles too. So left lateral ventricle, right lateral ventricle. It's located in it's in both hemispheres. It's located in all these lobes. I'm not going to test you on the different lobes, but I just want you to understand that it does go a long way. And so when we start seeing cuts of different sections of the brain, let me show you an example. So here's the front of the brain, the anterior view. We take a frontal section, we can see lateral ventricles. Take another section, lateral ventricle, but it's changing shape. Take another section, change the shape a little more. Take another section, change the shape even more, and so on. So yeah, depending on what region of the brain we're looking, it's going to look different because sections take just a two-dimensional view of something. Oh, okay. So lateral ventricles, largest ones on either side. The two lateral ventricles come together. See this? See how they come together like a Y? Like right lateral ventricle, left lateral ventricle, and then they have two little passageways that fuse and then form this one with a hole in it. Get my eye, there we go. So this one with a hole in it, that's the third ventricle. Third ventricle. So the lateral ventricles lead to the third ventricle. The third ventricle, shown here, it's located sandwiched. It's sandwiched right here. See that little hole here? This is brain matter. So because this is actual brain matter, that's why you've got something missing. That's why you've got something missing right here, because brain matter is going through it. This is the third ventricle. The third ventricle will continue on. We got a little passageway, and that connects it to the fourth ventricle. The fourth ventricle, you can see it in this mid-sagittal cut. That'll be right here. It's between this bulge of the brainstem, it's called the pons, and this back part here, the cerebellum. This space right here, that's the fourth ventricle. So lateral ventricles connect to the third ventricle connects to the fourth ventricle keeps going, connects to the spinal cord. In the spinal cord, it's called the central canal. Let me attempt to do a drawing over here. So we've got, I forgot to label this one. This is our arachnoid villus. Villus is singular, villi is plural. So here's an arachnoid villus. This would be lateral. Lateral leads to third. Third leads to fourth. And then fourth leads to central canal. So the ventricles aren't a place, they are passageways. A piece, a place, an eye. I mean, think of it this way: like if you walk through a hallway, that's a passageway, and then when you walk into a room, it's a room. So think of ventricles as rooms, but all the rooms are connected. So I can go from this room go through a hallway, go to the next room, go through a hallway, go to the next room. It's like that. So just the ventricles they open up a little bit more. Ventricles open up a little bit more. Is the central canal the same thing as the spinal cord? The spinal cord is the whole thing. The central canal is the hole in the spinal cord. Where's my model? So here's the spinal cord that's inside of the inside of the vertebral foramen. That tiny hole you see here, that's the central canal. The tiny hole is the central canal. This actually shows the dura. You can see the dura mater covering spinal cord, and then we can assume that pia mater is directly surrounding the spinal cord. But yeah, that tiny hole in the center, that's the central canal of the spinal cord. More questions? Just throw it in the chat, so I'll get to it. Okay. So like I just said, this is summarizing everything that I just described. All these ventricles, all these canals, the subarachnoid space, it's all one continuous thing. This really, really neat animation up here. You can see a pulsating, just like how blood pulses through our body, thanks to our heart. Cerebrospinal fluid is pulsing through our brain and spinal cord. We don't have a pump like the heart to move the CSF around, so it's moving slowly, but it's still moving. It's moving from the pressure created from the CSF produced by the choroid plexus and from its removal with the arachnoid villi. And yes, Andrea, the spinal cord has the same exact meninges. It's continuous. It's all one piece. So the way I drew it over here, it's all one continuous piece surrounding the brain and spinal cord. So if we follow the journey of cerebrospinal fluid as it's made, let's say we're starting in the lateral ventricles. We go from the lateral ventricles, the connecting passageway between lateral and third ventricle. You can see the third ventricle is sandwiched here in the middle. These are called interventricular foramina. Plural foramina, if you're just talking about one, it's a foramen. So lateral ventricle, interventricular foramen, third ventricle. From the third ventricle, we pass through the cerebral aqueduct. We'll be able to see this on our cadaver brain. This is the third ventricle surrounding this dot, third ventricle. This little passageway, that's the cerebral aqueduct until we get to the fourth ventricle. Cerebral aqueduct to the fourth ventricle to the central canal. We go down and out, make our way to the subarachnoid space, and then we can drain through the arachnoid villi to the dural venous sinuses. Just want to give you a heads up. This is the beginning of knowing pathways. So here's a pathway that cerebrospinal fluid takes. We'll be learning nervous system pathways. We'll be learning circulatory pathways. We'll be learning reproductive pathways. There's a lot of pathways. So get used to it. And I'll try to help you, of course. Just to relate that to things that you may know about, like you've heard of the word stroke, for instance. Sorry, let me back up. We talked about veins. Those are the dural venous sinuses. Veins drain blood away from the head. So what's delivering blood to the head? The two major groups of arteries that give blood to the head are the internal carotids and the vertebral arteries. You've seen the vertebral arteries before. Let me find the model with it. Where are you, model? It's over here. You've seen the vertebral arteries before. They go through the vertebrae. Can anyone guess what feature the vertebral arteries go through? Can anyone guess what feature of the vertebrae that the vertebral arteries go through? We can see a vertebral artery here in red coming up the side. I guess you can see them both here. So here's a vertebral artery going up, the vertebral artery going up. Anyone in the comments who can tell me what these vertebral arteries are going through? What's the name of the hole in the vertebrae? Yeah, that's right, Mackenzie. Transverse foramen, or plural foramina. These vertebral arteries are going through the transverse foramina in the cervical vertebrae, going up, delivering blood into the brain. Intervertebral is not the answer. Those peripheral nerves go through intervertebral foramina. Transverse foramina are going up and down. The carotids are more anterior. It's not on this model, but maybe it's on this one. The carotids are on the anterior part of the neck. I don't know how you can see this. See that red line there? Those are carotids. Those also go to the brain. You need lots of blood flow to the brain. The brain does a lot of stuff all the time. So internal carotids and vertebral arteries deliver blood to the brain. Blood leaves the brain through those venous sinuses, and then from the neck, the veins are called jugulars. So arteries deliver blood to the brain, veins take blood away from the brain. The jugular vein is draining blood away from the neck, head and neck. If you have a stroke, the technical term for it is a cerebrovascular accident. So it's a problem with the blood vessels in the brain, cerebrovascular. That's when you have a clot for one of several potential reasons. If you're blocking blood flow, that would lead to death. You could have an aneurysm. An aneurysm is when you have damaged blood vessel walls. You could have a buildup of plaque, buildup of tissue deep to the simple squamous of the blood vessels, and now it's blocking off the artery. You could have a clot. You could have damage of some other kind. There are several reasons why a stroke can happen. But if you block blood flow, brain cells are going to die. The tissue needs constant supply of oxygen and nutrients. Okay, yes, I know it was a lot. Let's take a moment to digest it. Take a moment. Andrea, I'm looking up your IA, acute ischemic. What does that mean? Acute means blood loss. Oh, acute ischemic stroke, AI stroke. That's acute just means like very sudden. Ischemic means blockage of blood flow. So it's just describing a type of stroke. Yeah, ischemic means it's blocked. Hemorrhagic would mean a blood vessel has been damaged and burst. So ischemic means there's a blockage. Where are we? Okay. People with a condition called septo-optic dysplasia. You have visual impairments due to distended lateral ventricles. Oh, you know what? I didn't go over this word with you all. The answer is B. My bad. Let me explain what this is. Okay, the term I forgot to introduce to you is called the septum pellucidum. Between the lateral ventricles, there's tissue. The lateral ventricles, they do connect with these interventricular foramina here, but they don't connect up here. There's a very thin wall. There's a thin wall between the lateral ventricles. The word for wall is septum. Remember, we went over nasal septum. Septum just means wall. Lucid, maybe you've heard of the word lucid. Lucid means clear. So it's a thin, clear wall separating the lateral ventricles. You can see it in a frontal coronal cut. This thin line separating the ventricles is the septum pellucidum. So that's in a frontal cut. In a sagittal cut, there would be a very thin tissue separating this is the lateral ventricle right here. There would be a very thin membrane, thin wall separating that. That's the septum pellucidum. Sorry, Emilio. Well, some of you figured it out, but yeah, sorry, my bad. Questions on that? The answer is B. Falx cerebri is separating the hemispheres, but doesn't go. The falx cerebri stops here. The falx cerebri doesn't go as far down as the ventricles. It stops right here. Choroid plexus is within the ventricle. Arachnoid villi are for draining cerebrospinal fluid. So yes, the answer is B. Septum pellucidum. Another question, and I did go over this. Okay, so we're blocking the passage between the third and fourth ventricle. Here's the third ventricle. Here's the passageway to the fourth ventricle. So this passageway, that is the cerebral aqueduct. Cerebral aqueduct. Central canal is down here. Interventricular foramen, that's connecting lateral ventricle to this one here. Lateral ventricle to third ventricle. Neural venous sinus, that's draining blood away from the head. Mackenzie, what is the structure called where the falx cerebri stops? You mean what it's attached to? In the anterior side? The falx cerebri would stop right here. This is the crista galli of the ethmoid. The falx cerebri is attached right here. We're not going to go over what attaches in the back, so don't worry about it. But it's a tentorium cerebelli. This is the crista galli of the ethmoid. Thanks, Corey. Okay, cool. Oh, one more question. And Mackenzie, you predicted this, so good job. All right, I see a slight mix of answers. We're talking about damage to the crista galli, like we just discussed. What attaches to the crista galli, and it's in the sagittal area? That would be specifically the falx cerebri. The falx cerebri is made of dura mater, but when the dura mater fuses together in the middle, in that longitudinal mid-sagittal fissure, it's falx cerebri. So it's C. C is the best answer. Because it's falx cerebri, it attaches, no, not this one. The falx cerebri attaches to the crista galli in the anterior side of the brain. Okay, it is 11:10. Let's take a break. I'm gonna end this feed. I'm gonna start a new feed for the lab. So look out for the new link. I'll start it here in a minute, so you can just hang out. I'll come back at 11:25. Come back at 11:25.